So our next presenter falls in that same category. When we were, when we were doing our work, and our staff was looking into different kinds of antemortem testing, live animal testing, particularly looking at some of the amplification techniques. Our next presenter's name popped up quite often. That is Dr. Rodrigo Morales. Dr. Morales obtained his Bachelor of Science degree in biochemistry in 2003 from the University of Chile, and a PhD in sciences from the same institution. His PhD work, which was performed completely at the University of Texas Medical Branch at Galveston, focused on prion strain and species barrier phenomenon in prion diseases and the transmission of Alzheimer's disease amyloid beta peptides. His postdoctoral studies were performed at the University of Texas Science Center at Houston, where he was appointed assistant professor in 2012 and associate professor in 2018. His current research interests involve the strain variation in interspecies transmission in prion diseases, the dynamics of environmental chronic wasting disease, contamination, the prion-like features of amyloid beta peptides, and the interaction of different amyloidogenic proteins as disease mechanism. Dr. Morales has authored more than 35 articles in the field of protein misfolding. Please help me welcome Dr. Morales.
Thank you very much for the invitation. And as mentioned, I will be talking about presymptomatic prion detection using an in vitro test that we call the protein misfolding cyclic amplification, or PMCA. I don't know, maybe you have heard about it. So this is the disclosure for my presentation. And I think that all of us agree that we are in an urgent need of a live diagnostic test for chronic wasting disease. And this is not a problem only for this disease, but for all other prion diseases, especially in humans and also in sheep, for example, and cattle. So one of the problems in order to find a good diagnostic test is, of course, to find an appropriate biomarker. And so far the most specific biomarker is the infectious prion, or PRPSC, that is what we call the prion infectious protein. So the problem is that this protein is in very little quantities. So in order to detect this protein in tissues or fluids, we have two strategies, right? One is to design more sensitive tests to detect the PRPSC in the tissues or fluids, or the other is to amplify the marker. So this work was started by my PhD mentor, Dr. Claudio Soto, who in the year 2001 described this protein misfolding cyclic amplification technique. So this technique, I will briefly explain a little bit how it works. It's based in the natural infectious process of prions. Basically you have the infectious material that we call again PRPSC, that when it gets in contact with the normal prion protein, or PRPC, it templates the misfolding and the aggregation of this normal protein into the misfolded isoform, or an infectious isoform. So this process is a very slow process, but with time will accumulate at a critical quantities in order to lead to disease. So in animals, in experimental animals, this can take months, and some other animals could take years, and in humans, as for example in the case of Kuru, that is a transmissible form of human prion disease, could take decades. So in order to accelerate this process, what we do in the test tube is to combine little amounts of the infectious agent of PRPSC with large quantities of the normal prion protein. So when we have them incubated in the tube, as I told you before, the infectious prions will template the recruitment, the misfolding, and the recruitment of the normal prion protein into the infectious particles. So the problem here is that this infectious particle grows only at the end. So in order to accelerate this process, what we do is to break them. We use sonication for this. And basically, when we apply sonication to the tube, we break these fibers that are formed. We create new ends that will accelerate the recruitment of normal protein into the misfolded form. And if we repeat this process in a cyclic manner, so basically we accelerate the process of protein misfolding that, as I told you, it could take months or years in animals to happen. So this process has been implemented in several animal species. This is just some example of it. So for example, we have done it in Syrian hamsters and in humans, and also in cervids. So I will explain a little bit more about this later. But I just want to tell you in here that with this technique, we are able to detect infectious particles from a brain that has been diluted up to 1 billion times. So the technique is really, really sensitive. And if we compare it to conventional tests, for example, Western blot or ELISA, we can see that the power of detection of PMCA is really, really great. So actually, estimations, mathematical estimations that we have made in the lab suggest that in some systems, like for example in Syrian hamster, we can detect up to 1 infectious particle. So the technique is really, really strong. So this technique has been utilized to detect prions, as I told you, in different systems. For example, we have done it in presymptomatic rodents, but also we have been doing it in humans affected by the variant form of Creutzfeldt-Jakob disease. These are the patients that have been affected by infected by mad cow disease. So I think that it's clear also what is the need of detection in prion diseases, but we are particularly interested in the laboratory in this and environmental contamination. So we are also particularly focused on white-tailed deer. One of the reasons is because PMCA is very specific in terms of the protein that we analyze. So for example, if we analyze prions from elk, the optimal for PMCA is to use elk substrate. And for example, if we use deer, the idea is to use the deer substrate. So we focus on this particular animal species, and then we are planning to move forward to other animal species. So work from several other researchers have shown that prion infectivity is present in several products derived from white-tailed deer. So we started to work on diagnosis using blood samples from white-tailed deer because it's a sample that is easy to get, and also it provides little chances for cross-contamination compared to other samples. And this is very important considering the power of detection that PMCA has. So this work has been done in strong collaboration with Dr. Tracy Nichols, who has provided us a lot of the samples that we have been analyzing. And when we are analyzing these samples, we need to consider some potential problems or variabilities in the analysis. One of them is that white-tailed deer present polymorphism in the prion protein. So as I told you, the PMCA is sensitive to these polymorphic changes, to the sequence changes in the protein. So the most important polymorphic variation in the prion protein is at position 96, where a glycine could be replaced by a serine. So the glycine at this position is the most prevalent in the deer population, and also is the one that is, provides more susceptibility to CWD compared to the serine counterpart. So the other variability that we need to consider is that these samples are not experimental samples. So actually, when we get the samples, we don't know when the animals got infected. So we need to stratify them somehow. So what we did, it was to stratify them as early presymptomatic or late presymptomatic, depending on the presence of the the infectious protein in retropharyngeal lymph nodes or brains. So when the animals got infected by peripheral routes, they first replicate in the periphery before reaching the brain. So when the animals show presence of PRPSC, of the infectious agent, only in the lymph node is when we call them early presymptomatic. But when the animals are advancing in the disease and the prion protein reaches the brain, then we will have positive signal in brain and lymph nodes. We will call them late presymptomatic. So in the first experiment involving blood that I will show you, this is a paper that we published last year. The first thing that we did was to optimize PMCA for white-tailed deer. Actually, PMCA have some variations between different animal species. We need to modify little stuff, like for example, the composition of the buffer in which we work, the cycles of incubation and sonication, etc. So this is, to make a long story short, Carlos, who was a PhD student at that time in the lab, he modified a lot of parameters on the PMCA to make it work in an optimal condition. And as you can see here, when we take a brain of an infected deer, we can amplify up to a dilution of 10 to minus 9. So when we take the brain and we diluted it 1 billion times, we can see amplification. And the maximum amplification was reached at the first round of PMCA, which means that we do it at 48 hours. So, and when we do serial passages, and serial passages mean that we try to replenish for, the reaction with fresh substrate to increase for sensitivity, we can see that the maximum amplification is reached at the first round. So basically what I'm wanting to say is that the PMCA is working in optimal conditions. So it's very good for detection, for potential detection. And in here are negative control samples that have no prions, you know, just to see whether there is some sort of false positives in our detection, and we cannot see anything. So the technique is working well. So then, in order to see whether this technique was suitable to be used for blood detection, what we did was to take blood from healthy deer and we spike it with different dilutions of a brain extract from a CWD infected deer. And as a control, we use the same dilutions of CWD, but in buffer, in PBS. It's the buffer that we normally use, it's like the normal condition. So in here, you can see that the blood, the presence of blood in the reaction kind of inhibit the PMCA. We have no detection in the first passage compared to the buffer situation, the normal situation. However, when we do serial rounds of PMCA to increase sensitivity, we reach in both cases to the same limit of detection. So basically, this is telling us that even though the technique is working very well in the first round, in order to have a good detection for blood samples, we should do more rounds of amplification to increase the sensitivity to kind of go away of this inhibitory process that we have for blood. So when we had that data, first we received from Dr. Nichols some samples that were not blinded to us, actually were samples from animals showing poor body condition, and from animals also that were retrieved from CWD-free farms. So in order to test, the idea in here was to test the specificity of the technique. And as you can see from these samples that were coming from CWD infected animals with showing poor body condition, we observed a good detection, and from the control animals, we didn't see anything. So it seems, at least from this small cohort of samples, that we have 100% sensitivity and 100% specificity for the test. But then the question was what happened with more real samples from pre-symptomatic animals, animals not showing clinical signs. What happened if we can detect prions or not? And we received a bunch of samples, 87 actually, and 13 negative controls, but at the moment of receiving the samples, we were blinded to them. So the identity of the samples was revealed after we provided the data. And from this group of 87 samples, 34 were at the early presymptomatic stage, and 48 were at the late presymptomatic stage. So this is a sample of the of the test that we used. It was, this analysis was performed by two independent investigators at two different rooms. You know, everything was completely done independently. They got similar results on them. And we observed that the detection of prions in pre-symptomatic cases of CWD was 100% specific and highly sensitive. We didn't detect a prion signal for any of the negative samples. But in here in the table, we can see that we observe some variability depending on the stage of the incubation period the animals are. For example, for the asymptomatic animals at late stages of the incubation period, we observe a correct diagnosis in 96% of the samples. For the animals at early stages of the incubation period, we observe a detection in 53% of the sample. So, in summary, we obtain, again, 100% specificity in the test and approximately a 78% sensitivity, considering the type of samples that we got, because if we got more asymptomatic samples at later stages, maybe this number could have been switched. So it is important to mention that for this analysis, we use only the predominant prion protein polymorphism, the one expressing glycine at position 96. So, as I mentioned, PMCA is sensitive to these to these sequence changes in the protein. So, again, we use glycine at position 96 for the animals from where we retrieve the samples, but also as a substrate for the PMCA. So the question is what happened with the detection in these animals showing the other polymorphic variation at position 96, either showing glycine and serine or only serine at this position. So in here we receive another set of samples from where 26 were glycine or serine at position 96, and only 5 were serine and serine at position 96. Now, for this experiment, it's important to mention that we use as a substrate the glycine version of the substrate. So we expected some sort of incompatibility on the detection. However, it was the only substrate that we had at the moment. So when we did this analysis, we observed that for the late presymptomatic, we had for the GS approximately 60% of the animals with a correct diagnosis, and for the early presymptomatic around the same number. So this is kind of strange for us, because we will expect that at the time that the animals are advancing in the incubation period, they should, in the incubation of prions, they should be displaying more prions in the blood that could be more easily detected. However, we are pending to analyze this sample in the other polymorphic group using the different substrate and see whether we can increase the sensitivity of detection. And unfortunately for the SS group, we didn't have so many samples. However, we were able to detect in 1 out of 5 of the early presymptomatic. So overall, the detection in the GS and the S blood samples was 100% specific. Again, we didn't have any false positive, and was 51.6% sensitive, considering all the samples that we analyzed. As I told you, the lower efficiency of detection may be to the substrate and infectious agent incompatibility, or could also be that these animals expressing this polymorphism have lower prions circulating in the periphery. So this is something that we are currently investigating. And as I told you, experiments using the homologous PMCA substrate are currently ongoing. So another set of samples that we analyzed was semen and male sexual tissue. So for that, we also got samples from Dr. Nichols, and the samples that we got here were also blinded to us. The identity, the CWD status of the animal was revealed only after we had the data. So it is important to mention that it has been detected prion infectivity in semen from scrap-infected sheep. Actually, this was identified also using PMCA. However, the data is some sort of contradictory, because even though there is presence of, it may be presence of prions in semen, maybe it's not enough to produce infectivity. So this is an important question, I think, relevant for CWD, whether one thing is that whether we find presence of infectivity in semen samples, but the other is whether the amount of prions present in semen, if they are, is enough to produce infectivity. So for that, as I told you, we received some samples that were blinded to us. Unfortunately, they were not too many, but these type of samples are very difficult to obtain. And we obtained seminal fluid, tested stroma, and epididymis. So this is the result of the PMCA we have. So these are the Western blot after the PMCA. It's better illustrated in here in the table. So for the semen, we again stratified the sample as late presymptomatic or early presymptomatic. For the late presymptomatic animals, we were able to detect in 100% of them. So this is very important because the animals were not showing any clinical signs. But after post-mortem examination, they had presence of PRPAC in the ovex and also in the retropharyngeal lymph nodes. But for the animals showing prions only in the retropharyngeal lymph nodes, we couldn't detect in any of them. So for testes and for epididymis was something similar, although for the early presymptomatic, we were able to detect in a low proportion of the animals that we analyzed. Very little samples, I told you, to analyze, so we couldn't make a clear correlation between the different polymorphic groups and how this polymorphic variation affects the presence of prions in semen or male sexual tissues. However, from this small group of samples, we may say that there is no major influence. So it seems that the prions have a tropism to these fluids and tissues at the late stages of the incubation period, so it doesn't occur at early stages. And regarding the specificity, so the specificity was very good for semen, 100%, for testis also no false positive, and for epididymis, unfortunately, we had one sample that was constantly detected as positive. We did the samples, I remember, several times, and it was very consistent. So I think that this is something that it could be expected considering the nature of the collection of the samples. I think that we will be hearing something later about that because these are field samples, and chances of contamination are present. And also considering the power of detection of PMCA. PMCA is very sensitive, so if there was a slight contamination of it, the technique will catch it. So eventually, if this technique or other similar techniques with similar power of sensitivity are used for detection, collection of samples is an important factor to be considered. So as a conclusion of this slide, CWD prions are detected in semen and male sexual tissues of CWD-infected deer, and detection is high at the late stages of the CWD incubation period. So saliva has been shown to show high infectivity titers compared to other bodily fluids, and we wanted to analyze also that in field samples, again in collaboration with Dr. Tracy, who provided us some samples. We did a small, relatively small, it was about 100 samples that we analyzed. So as you can see, this is our control, it's just to see how the PMCA was working, and these are the samples, and you can see only one sample detected over there. At the beginning, these samples were blinded to us. And when we got the result, we were very disappointed. We said, oh, the technique is not working. We're supposed to be detecting a lot in saliva. But then when we got the result from Dr. Nichols, we saw that that was the only positive sample. So actually, it was a good test of our technique to show the specificity. So we didn't have a false negative. So it was only one animal positive, and we were able to detect it. So again, this is white-tailed deer, and in here, I am just showing you that using the same parameters that we were using for detecting in here, we were able to detect prions in saliva of CWD-infected elk. So in here, as you can see, it took us 3 rounds, so it took us longer time to detect the prions, but also you need to consider, as I previously mentioned, that we are using a heterologous system. So we were using deer substrate to detect elk prions. So we expect that if we use the elk substrate, so the ELK normal protein, we will be detecting it much faster. So CWD detection in saliva samples is possible in presymptomatic animals. So we can see here in this single sample, and this needs to be tested in a higher number of animals, in more representative samples, to see whether we can use it for saliva. Another samples that we are analyzing, also feces. In this set of samples, they were not blinded to us. Also, it was to prove whether our technology was useful to detect prions in feces. So Dr. Nichols sent us feces samples from field-collected samples, actually, from animals at the again, late presymptomatic stage or early presymptomatic stage. We tested the different protocols to treat the feces, because if you put the feces directly in our reaction, it messes up the reaction doesn't work, it has a lot of inhibitors, so we need to prepare the sample before analyzing. And when we do that, we found, this is an example of two protocols that we found they were the ones that were working better. And as you can see, for example, we didn't have any false positive. Again, all the CWD negative samples were negative. For the animals at the early stages of the incubation period, similar scenario to what we had for semen, no detection. But at later stages, we were able to detect in 5 out of 6 or 6 out of 6 samples. So it's looking promising. And now we are analyzing a much larger set of samples from which we don't know what it is. So it's blinded to us. So CWD detection in feces samples is possible for presymptomatic animals, and however, the detection seems to be less efficient compared to the blood test. I have just 5 minutes to finish, so I will go through this very fast, but this is the cycle of events that we believe that is occurring in CWD. So one important component that we believe is going from the infected animal to get a healthy animal infected goes in the environment, and how prions accumulate in the environment. So as previously mentioned, we published a paper, it was in 2015-16, about the contamination of prions in plants. So we found that we are expanding this experiment now, but this paper that was published early this year deal with how prions can differentially contaminate different materials, and how these contaminated materials could act as vector of disease transmission. So to make the long story short, we used this hamster strain. Hamster is experimental model, it's not deer, I know, but it allows us to work in the lab in a fast and cheapest way. We cannot have infected deer in the laboratory, so we can have the hamster infected in the laboratory and model how the disease could be transmitted by contaminating environmental components. So when contaminated different components, we observe that different materials have different abilities to bind prions and to transmit disease. So, but also we did it with prions from other species, including mice, humans, and actually deer. Something that may be interesting for now is that there was some materials that consistently show a low ability to transmit disease, like for example brass, and now we are expanding it to copper. We are analyzing more materials, but I don't know, maybe it will be preferred for some practices to avoid some materials and to prefer some others, because some materials could enhance the ability of transmission and some others could avoid it. So after infecting the materials, we transmit into animals. So we observe similar results. For example, brass was the one showing extended incubation periods compared to all the others. And when we contaminated the spheres made by different materials and we place it in the cage of the infected animals, and just to see whether they get infected by contact, just by contact, we observe actually again that brass was the one that was the less successful to transmit the disease. So to conclude, our PMCA protocol allows for the replication of white-tailed deer prion detection with high sensitivity and specificity. We tested in a different type of samples, including blood, saliva, and feces. We also are showing, I believe, for the first time that prions are present in semen and male sexual-related tissues of CWD-infected deer. And the tropism seems to occur at late stages of their incubation period, although we are pending to see how much is the quantity of infectivity they have. Also, we show that prions can attach to a wide variety of surfaces. We mostly did this experiment using hamsters, Syrian hamsters as a model. Now we are doing it on CWD, specifically for white-tailed deer prions. And the PMCA technology can also be used, as I showed you, to detect prions in environmental components, including plants and soil. And, well, as I told you, further infectivity experiments in transgenic mice and white-tailed deer will clarify the role of sexual transmission and parasites and other environmental components for the transmission of CWD. So just to conclude, most of the work was done by a former PhD student, Carlos Cram, who is finishing his thesis now. This guy, Ruben Gomez, also participated in some of the studies. Of course, to thank Dr. Tracy Nichols for all the samples that, invaluable samples that she provided to us for doing this experiment. And also Dr. Glenn Telling from Colorado State University, who provided the brains for doing the PMCA. And thank you for your attention.